Good evening, everyone. Thank you for being here. My name is Amanda Thomas, and I would like to welcome you to Science on Tap. We are here on a Wednesday night for a special talk on supercomputing and systems biology, a new hypothesis for COVID-19, with Dr. Daniel Jacobson, who is the lead researcher and chief scientist for computational systems biology at Oak Ridge National Laboratory. And I'm very excited to have him here. This is many of you may have seen this. The way I found out about this topic was that when a dozen or more of my Facebook friends posted a link to this article. And it was, some of you may have read this, some of you may be seeing this for the first time, but I was interested because it describes some data that may give us a new understanding for how COVID-19 works, and some possible treatments that are already FDA approved. And another thing that excited me about this is that it's an example of showing real-time science, and a real-time example of how science works. And they are talking about science-based data that they have collected and analyzed, and that has been vetted through a peer review process. And that's how science works. It doesn't just show up on a YouTube video somewhere. So I'm glad to have an opportunity to talk about something that is real science, and potentially very exciting. So we'll see. And as luck would have it, I read this article and then found out that I know that I have a friend who works at Oak Ridge National Labs, and he put me in touch with Daniel. And so, hi, Jared. Thank you. And here we are this evening. So, and where are we? We are at Science on Tap. As I mentioned, we're doing this on a Wednesday night. Normally, we've been hosting our events on Thursday, but we were able to get Dr. Jacobson here this evening. Science on Tap is an event series that has been running in Portland, Oregon and Vancouver, Washington for about 7 years. And until a couple of months ago, we were doing live in-person events, and now we're doing live online events on Thursday nights, most Thursday nights anyway. Our goal is to make science fun and accessible to adults in particular. And as true to our name, if you would like to enjoy a tasty beverage while you are listening this evening, I encourage you to do that, whatever beverage suits your choice. What you can expect this evening is that our speaker is going to talk for about 45 minutes, and then we'll have Q&A. I have a team of people who are watching the comments and the chat, and they will be sending questions to me. I will come on at the end and we'll ask as many of those questions as we can get to at the end of the presentation. One quick note is that Dr. Jacobson is not a medical doctor, so don't ask any questions about medical things because he's not going to be able to answer those. And so, yeah, just a little caveat there. If you are watching on Zoom, you can write comments in the chat, and you can chat in the chat with other people, and you can also write questions in there. You can also write questions in the Q&A if you're watching on Zoom. If you're joining us on Facebook Live, you can put those in the comments, and we have somebody who's monitoring that as well, and everyone will send those comments to me, and I'll ask those questions at the end. And we are recording this, and this should be available for watching on YouTube in the next day or two. So with that, I'm going to stop sharing my screen and stop talking and welcome our speaker to the stage, Dr. Daniel Jacobson.
Thanks, Amanda. I'd like to talk a little bit about tonight of some of the COVID-19 work we're doing, but first, by way of introduction, I'm a computational systems biologist at Oak Ridge. And we're really interested in trying to understand all the combinatorial interactions of molecules and cells that lead to how an organism functions, how does it work, and that lead to traits or phenotypes, or in this case, diseases. We do this with a broad range of species and a wide range of projects. We do everything from bioenergy to ecosystems work, all the way through to model organisms and biomedical studies. And the biomedical side, we do a lot of neuroscience and substance abuse work, as well as other areas as well, like cardiovascular disease. So we're involved in a lot of different areas. And we often find that the algorithms and approaches that we design for one area works perfectly well in another. And so we see a lot of integration possibilities across our lines of research. So as a brief sort of reminder of the central dogma of biology, we'll be talking about different omic layers tonight. So the genome, composed the DNA, really sort of the blueprint of the cell, and how that's actuated into transcribed into RNA, and how RNA is then later translated into proteins that often are the workhorses in the cell. We'll be talking a lot tonight about RNA, about gene expression, and how differences in gene expression can change the outcomes, in this case, in disease. Now, the full story is more complicated than this. You have epigenetic mechanisms, you have post-translational modifications, you have metabolic interactions, but this sort of central simple dogma, and we'll focus a lot on really gene expression tonight, along with a few other things, as well as a lot of clinical data as well. We tend to view the world as these different layers of data. So if we can convert layers of data for small sets of samples or for whole populations into matrices, then we can find patterns within and across those matrices, whether that's at the genome level, whether that's at the RNA or transcriptome level, so gene expression, whether that's at the protein or proteomics level, the metabolites or metabolomics level, or phenotypes, these outcomes of all these interactions within and across all these layers lead to the traits of an organism. And when things go wrong somewhere in that chain, you get altered phenotypes, or in fact, disease states. And we're also interested how all of those complex interactions are conditional on environment. And when I say complex, the combinatorial complexity of a single human genome in a human cell is on the order of 10 to the 170. That's a lot of zeros. If you think about the number of stars in the universe, it's around 10 to the 27 or so. If you think about the number of atoms in the universe, it's about 10 to the 80. So when we talk about biological systems as complex, they're enormously complex. And we're trying to understand not just the possible interactions, but the interactions that actually happen within and across all of these layers of functional elements in a cell, and how that all builds up across cell types and tissues and organ systems into fully functioning organisms, and how that's all affected by these different changes. We capture these relationships with a range of different computational approaches and statistical approaches. Often now data analytic, and especially AI, explainable AI, machine learning approaches that will capture a lot of these relationships. We've done a lot of work with new algorithms that can capture in a sophisticated way these sorts of relationships. And then often store them as networks. The networks are simply graphs, so you have nodes and edges. The nodes you can represent really any biological object, and the edges are just representing relationships between those biological objects. Once we have that representation of a complex system, we write a lot of algorithms that can then interrogate those network models and tell us the biological narratives that they're representing. So tonight I'll be talking about work we're doing both in humans for pathogenesis, and a little bit about what we're doing in coronaviruses themselves, and how we're combining all of this information, not just in network models, but also looking at the three-dimensional structure level. I'll touch on that briefly, but we're doing a lot of work in my lab to develop very sophisticated pipelines that can predict the structure of proteins just from the primary sequence. And we're getting better and better at doing that at scale. So there's an interesting set of capabilities that we can not just represent interactions, but we can start to look at the three-dimensional interactome. And then, of course, we're interested in how small molecules are going to interact with those proteins. Especially for COVID-19, we're focusing on existing drugs to see if they're existing therapies that we can repurpose to help treat COVID-19 patients if they prove out through clinical trials. We've spent quite a bit of time thinking about the life cycles of coronaviruses, both historically what is known about previous coronaviruses and what can we learn about this new coronavirus, SARS-CoV-2. I think we've all probably heard about the ACE2 protein is certainly one of the proteins that this coronavirus latches onto to get into the cell. The SARS-CoV protein virus also used this ACE2 protein and possibly the ACE protein as well. It's a way to get inside the cell. Once it's inside the cell, it's really amazing what this sort of bag of 27 proteins, the tricks it has up its sleeve. Those proteins go off and interfere with and re-engineer effectively the machinery inside the cell to do the virus's bidding rather than the human cell's bidding. One of the effects that we have a strong hypothesis about is that one of the proteases, the 3CL protease from this current coronavirus, actually interferes with the activity of a transcription factor called NF-kappa B. Transcription factors are proteins that actually regulate the expression of other genes. And so one of the papers that will actually come out either this week or next week talks about our work predicting that interference. Keep that in mind. We're going to come back to NF-kappa B as a key regulatory element that the virus perturbs that in many cases may lead to the pathogenesis, may lead to all these symptoms that we're seeing in COVID-19 patients. As I mentioned, we're doing a lot of work with gene expression data. So we've looked at a population level gene expression data of 17,000 samples across 57 different human tissues. We've also looked at single cell RNA-seq data sets. So here we're actually getting gene expression profiles from individual cells. It's a relatively new technology that allows us to see not just what's going on in a tissue, but within all the cell types within a tissue. And then we're looking at the gene expression data from the internal luminal surface of the lung in COVID-19 patients versus uninfected patients. What we're seeing here are the single cell results of ACE2, this well-known entry point for SARS-CoV-2, and another gene in that same pathway, ACE. Both of these are in the renin-angiotensin system, which is largely responsible for regulating your blood pressure. So this is a profile of where these genes are expressed in different cell types. And we see there's quite a bit of ACE2 expression in nasal samples. So it's pretty clear that an entry point for this virus is through the nasal passages, and it seems to colonize there. That's where most people are tested for the virus, is in those nasal passages. There's some evidence from mouse single cell data. There's a fair amount of ACE2 expression on the tongue. So the oral cavity is another place the virus can get a foothold. But interestingly, as we get into respiratory cells down in the lung, there's often very little ACE2 expression originally. There's a few sparse cell types that have some ACE2 expression. But it's an interesting question of whether infection moves directly from the nose down into the lungs or if there's a more circuitous route. So let's look at that a little bit. If we now look at the population scale expression data, so these are gene expression measurements for about 40,000 genes measured across about 1,000 individuals in all those 57 different tissues. And we can start to get a sense of the distribution of these genes, so where are they normally expressed and at what levels, and how does that vary across the population. So we see for ACE2, this gene that the coronavirus uses to get into cells, it's actually very heavily expressed in the intestines and the small intestines and in the testes, and lightly expressed at some other places at lower levels. It's actually quite, again, lightly expressed in the lungs. It does have some expression in the brain and, of course, nasal cavities. And it's fairly omnipresent in blood vessels as well. We can also see there can be tremendous variation across the population. Some people have much higher levels of expression in the lungs or the heart or the thymus. This may be part of the explanation why some people are more susceptible to the virus. And if we look at the other gene of interest, ACE, we see that it's actually quite highly expressed in the lungs, and even more so across the population, also very highly expressed in the small intestines and testes, but generally higher levels. So we'll keep an eye on ACE for a minute. It turns out to be an interesting part of the story. Now, after looking through all these tissues, we came up with some new hypotheses for different routes of infection. So as I mentioned before, clearly coming in through the nose and colonizing nasal passages is one way. But what happens from there is a little bit of an open question. Does it migrate down directly into the lungs? Or does it migrate down the esophagus and into the stomach? This coronavirus is incredibly pH stable. It can survive very low acidic pHs in the stomach, and it can also survive really high alkaline pHs. So it's actually doesn't really care about the acidity of the environment it's in. So we think it passes just fine through the stomach, and then probably colonizes the small intestines where all those ACE2 receptors are, and that becomes a reservoir for infection. From there, it can either enter the bloodstream through portal circulation, or it can also enter the lymphatic system. And in autopsies, we have certainly seen clear evidence that it is in the lymphatic system. Once it enters the lymph ducts, it will make its way up through the thoracic duct, which actually dumps into the bloodstream right above the heart. And from there will be pumped directly into the highly vascularized lung tissue, hoping to be oxygenated. So it's quite possible that there are multiple ways for the virus to get to the lungs and other organ systems. It may come in through the air passages, but it may come in the back door here, actually through the circulatory system. The terminal ends of your lungs are alveoli, these small little air sacs, and they're covered with capillaries that are responsible for gas exchange, releasing CO2 and taking up oxygen. So if we clearly have both nasal and respiratory shedding, but we also know from a range of data sets that there are viral particles being shed in feces. We also know that when you flush a toilet, the part of the contents of that toilet aerosolizes. You will have this viral plume coming out of a toilet if somebody's shedding virus in their feces. Keeping in mind that virus can live on plastic and metal surfaces for 4 days and up to a day on fabric surfaces. That means we have to be thinking about bathrooms in hospitals as well as public places as possible mechanisms of infections. And in fact, some studies have already been done looking at doing swabs and aerosol tests in bathrooms and hospitals, and find that they're really hot spots for viral particles in the bathroom. So that's another mode of infection we need to be concerned about. It's also turned into an interesting way to monitor the virus. Increasingly, cities and now even universities are monitoring viral loads in the population by sampling the wastewater, sampling the sewer system. I know there are universities that are sampling the wastewater coming out of each individual dormitory on campus to try to get an indication of what dorms may have sick students. Now, a lot of our mechanism came out of analysis of these bronchoalveolar lavage samples. So BAL samples are inserting a bronchoscope down into the terminal reaches of the lung. You really wedge the bronchoscope as far down as you can go, and then you inject sterile saline in all the way down to these luminal surfaces, getting all the way down, hopefully to the alveoli, and then you suck it back out. And you do that about 15 times, you get a very good wash at these terminal branches of the lungs where gas exchange is going to be occurring. In doing so, we're going to be picking up the cells that are on that surface, inner surface of the lung, any viruses that may be there as well. And we're really interested in what's happening, how those cells are responding to the presence of the virus. We've done this for COVID-19 patients and for looked at a data set of uninfected controls from actually a different study. But together that gives us a comparison we can do to see what's changing upon infection. Now, one of the systems we've been looking at, as I mentioned, ACE and ACE2 are in the renin-angiotensin system. This is primarily responsible for managing your blood pressure and fluid balance. But interestingly, it's often targeted as receptors for a whole range of different species of coronaviruses and humans and other mammals as the way that they get into cells. So sometimes it's ACE or ACE2, sometimes it's other members of this pathway, but it's often a member of this pathway. So we've been staring at this pathway in our molecular evolution of the virus studies for quite some time. And you sort of see right off, there's this dynamic balance in this square here between ACE2 and ACE. You can imagine that if ACE is downregulated, you're going to tip this balance of the peptides in the pathway. The entire pathway is really formed of proteases that are cleaving proteins down into different forms of peptides that then will go on to bind to receptors and have an effect on the cell. And the sorts of peptides you're gonna get really depends on the flow through the pathway. So if you downregulate ACE and leave ACE2 the same or upregulate ACE, you're gonna start to tip the balance pretty quickly towards Ang-1,9. And Ang-1,9 is an interesting set of these angiotensin peptides, and it only binds to the AT2 or the ATGR2 receptor, and that will become important in a second. But interestingly, if you remember, we talked about NF-kappa B, that this transcription factor that the virus targets right off the bat, it's a key regulator of ACE. And so we see the virus going after and trying to destroy the regulatory elements of NF-kappa B, and we immediately wonder, is that going to have an effect on ACE levels? So we looked in the gene expression data of what's going on in this entire pathway, and what's different between healthy individuals and COVID-19 patients. To better understand what we were looking at, we first took all of those, that large gene expression study at population scale, and here's where we use Summit with a number of different, both data analytic and explainable AI algorithms to understand the regulatory relationships. How are these genes expressed together or differently? How are they co-regulated, and what function can we infer from that? And very quickly we saw this axis between the two, this dynamic tension between these two receptor pathways with ACE on one side and ACE2 on the other. And we also started to see evidence of co-regulation with genes in the bradykinin pathway and downstream receptors, and functions involved in immune function, inflammatory responses. So immediately out of this network model of showing how things are normally co-regulated, we can start to infer some of the patterns to watch out for and some of the functions that may be affected. So let's look at how gene expression is changed in these COVID-19 patients. And this is a representation of both the RAS pathways as well as the bradykinin pathways. Let's actually start at the top. Coming into the first stages of the RAS pathways is a gene that, almost the first step, the second step, called renin, that's processing angiotensinogen into angiotensin 1, which is still an inactive peptide. But regulating that, very first step here is the vitamin D receptor. We noticed immediately the vitamin D receptor is a negative regulator. It's a repressor of renin. So higher levels of the vitamin D receptor will mean lower levels of renin. We see that the vitamin D receptor is downregulated. The color key here is blue is downregulated and red is upregulated. And these are log-fold changes, so quite large changes. So with vitamin D downregulated, we also noticed that genes that are normally responsible for catabolizing, chewing up, degrading vitamin D and its important metabolites are significantly upregulated. So we would predict this would lead to a big drop in vitamin D levels. So there's even less vitamin D to bind and activate the already downregulated vitamin D receptor, which will result in much more renin expression. And that's exactly what we see, is a boost in renin expression. If we keep going through the pathway here, you see this sort of branch between ACE and ACE2. Well, ACE, as we predicted from the NF-kappa B results, is severely downregulated. So we're going to be producing much less angiotensin 2. We're going to tip the balance towards angiotensin 1,9. This is important because that will bind the AGTR2 receptor And one of its jobs is to resensitize the bradykinin receptor. Normally the bradykinin receptors, when they see too much bradykinin, they actually downregulate. They say, you're talking too loudly, I'm gonna listen. They plug their ears, they stop listening to the signal. This receptor forces them to keep listening, forces them to keep responding. So okay, let's look over at the bradykinin pathway. What's happening at the very top? And the C1 inhibitor protein encoded by SERPING1 normally, again, is a negative repressor at the very top of this pathway. So something that inhibits this pathway from starting off is severely downregulated. So it sort of opens the floodgates in the kallikrein pathway to start the cascade that will lead to bradykinin. We also see a couple of the genes in the kallikrein pathway are upregulated. So that's even going to increase more synthesis of kallikreins through the pathway until we get down to bradykinin. And then the conversion to DesRG, an alternative form of bradykinin that can bind a secondary bradykinin receptor. So here we now we're going to have an out of control cascade of bradykinin. We've taken the brakes off of the system. Another key element of this is normally in the body, it's actually the ACE protein that degrades bradykinin itself. It's another one of these checks and balances. It's actively trying to get rid of bradykinin. So downregulating the ACE gene, having it under stress means bradykinin is being synthesized and not degraded. So this is what we're calling the bradykinin storm. The bradykinin's out of control. We've taken all the brakes off the system. We've forced the receptors to keep listening to the signal, and this will have effects downstream. This will activate phospholipase A2, which is the beginning of the arachidonic acid pathway, which has downstream signaling effects, which affect the permeability of your blood vessels and affect inflammatory responses. So an excess of bradykinin will cause actually gaps to open up in your blood vessels. And so fluid and plasma will start to flow out into the surrounding tissue. And immune cells that are normally constrained to the bloodstream will start to leak out into the surrounding tissue. And that's a classic inflammatory response. And that's all regulated by bradykinin. So now we have this hyperpermeable set of blood vessels. The internal surface of your lungs is lined with lots and lots of capillaries. You have about 70 to 100 square meters of surface area inside your lungs, all packed with these capillaries trying to exchange CO2 for oxygen. But now they are all becoming very leaky, and fluid is going to be flowing into the surrounding tissue. So you start to see a lot of fluid in the lungs of COVID-19 patients. Now, it gets worse than that, is that there's another natural molecule being produced in the lungs and various places in the body called hyaluronic acid. This is actually a fascinating biopolymer, the series of carbohydrates that can get quite long and large. And we find it all throughout biology. We find it in bacteria produce it, that sort of sliminess of a bacterial culture. Some of that is hyaluronic acid. Snail slime, that slimy mucus in a snail trail, a lot of that is hyaluronic acid. It's normally produced in low amounts in the interior surface of your lung as sort of a protective layer. It often protects against bacterial infections. It also serves as sort of a lubricant for mucus to be moved out of your lungs by the cilia. But in COVID-19 patients, we see the genes that are responsible for synthesizing, and all three of them are significantly upregulated, which is what these big red arrows are representing. So you're overproducing, you're over-synthesizing hyaluronic acid. And it so turns out that the genes responsible for degrading hyaluronic acid are severely downregulated. And there's a connection through to the RAS pathways directly, in that we know that the downregulation of ACE, we keep coming back to ACE, actually leads to this decrease in angiotensin II, which leads to the decrease of these two key enzymes for degradation. So exactly what we were talking about before in the RAS pathway is shutting down the degradation of hyaluronic acid. So again, the checks and balances are way out of whack. We're over-synthesizing this and not degrading it anymore. Well, why does that matter? Well, it turns out that hyaluronic acid is not just a polymer. It can become a very potent hydrogel. It can absorb over 1,000 times its own weight in water. when hyaluronic acid hits water, it swells up like a gelatin, like a jello. So you can imagine in a severe COVID-19 patient, and actually even in asymptomatic patients, we see this overproduction of hyaluronic acid. You're starting to leak fluid into your lungs, and that turns into basically jello in your alveoli. And so normal oxygen and CO2 exchange going through these alveoli, this air sac, where the capillaries are very near the surface, the air can flow through, oxygen will be absorbed, CO2 will be released. But when you fill up your alveoli with this hydrogel, the gas can't exchange. It sort of bounces off. It can't diffuse through the hydrogel to get down to the capillaries to do gas exchange. And this explains one of the reasons we have such a high mortality rate in ICUs and trying to ventilate patients. At some level, it doesn't matter how much oxygen you pump into their lungs, it simply can't diffuse through the hydrogel. You've probably heard about the radiological findings, the chest x-rays where they talk about ground glass opacities that look something like this. Well, we've looked at other diseases that overproduce hyaluronic acid in the lungs, and they have the exact same phenotype. You see ground glass opacities in the x-rays of patients with big increases of hyaluronic acid. So from other diseases, we can make the link that it's probably hyaluronic acid, this exact phenomenon we're describing, that's causing these radiological findings in COVID-19 patients. Now, alarmingly, there was a study that just came out a week or two ago taking chest x-rays of asymptomatic patients. So patients that are otherwise feel fine, they have these same ground glass opacities in their lungs. So they may not be aware of it yet, but this damage is being done. And there may well be a tipping point where it builds up enough and where all of a sudden they're not getting enough oxygen and they crash. And that has been another feature of COVID-19 of people seeming to be fine and very quickly going downhill. That may well be the case, but that's also alarming because asymptomatic patients having damage being done may have long-term consequences that they're not aware of. So from a public health perspective, that's something we should be very cognizant of. Now, if we project this model into other tissues, so we know if this model is happening in the lungs, we know that this virus has what's called tropism. That means it can infect many different organs, many different tissue types. If it has the same effect in those organ systems, what's going to happen? What happens if there's an excess of bradykinin, if there's a bradykinin storm in your muscle? Well, we know from other diseases that will give you tremendous muscle pain and soreness and joint pain. That's exactly one of the big symptoms we see in COVID-19 patients. We know from other diseases an excess of bradykinin will lead to encephalopathy, swelling of the brain, dizziness, headaches, cognitive impairments, brain fog, ischemia. That's caused by this fluid leaking into the surrounding tissue. And there have been several studies doing MRIs of brains showing exactly that. There's a lot of fluid leaking into the brain and putting pressure on the brain itself. We've talked about this pulmonary edema and hyaluronic acid, the vasodilation and hyperpermeability that will lead to low blood pressure, hypotension, and all the inflammatory responses. These big shifts in bradykinin will also lead to big shifts in electrolytes, and importantly, potassium. Potassium is a really key metabolite or electrolyte involved in heart function. So if you have big shifts in potassium, that can lead to arrhythmias and sudden heart failure, also symptoms we see in COVID-19 patients. As we keep marching through the body in different organ systems and keep looking at the list of symptoms we see in COVID-19 patients, we keep seeing matches between what we would expect to see from excess bradykinin and what we're finding in COVID-19 patients. It also helps explain why there's such a diversity of symptoms in different patients. If this is a local effect at the point of infection, then you're going to get different symptoms based on where the virus is infecting, what organs it reaches in your body, and that may be different in different people. That may be genetic or that may be stochastic, and may be random as to different tissues it actually colonizes, and that will explain this diversity of symptoms. Now the good news, that's all the bad news. Now the good news is, if we look back at this pathway, there are already 10, actually about a dozen different drugs we've identified already that can correct parts of these mechanisms that they're going wrong. There's a study that just came out of Spain last week about a vitamin D analog that's showing really encouraging results. So if you can break this regulatory chain and stop overproducing renin, you can stop some of the bad downstream consequences. There are drugs that can correct this decrease in SERPING1. There are drugs that can inhibit bradykinin synthesis at this KLKB1 gene. And there are drugs that can block the bradykinin receptors to prevent bradykinin from binding, to effectively inactivate these receptors. And there's also, some of you have probably heard about the recent results with dexamethasone, which is a corticosteroid. That attacks phospholipase A2, which is the gene directly activated by the bradykinin receptors that leads to the release of arachidonic acid. There have been 3 studies already, a vitamin D study that's showing encouraging results, efficacy. There's been a study in Europe on an antagonist, a blocker of the bradykinin receptors, incavidant, that's showing encouraging results. It's on small scale. We need to do much larger studies, but it seems it's providing patient benefits. And then dexamethasone, 7 studies recently have shown it to be effective. So we have 3 different points in our model that pharmaceutical treatments seem to be confirming, seem to be adding more lines of evidence to support this model. Downstream, again, we see NF-kappa B is involved in ACE regulation, and we know that's downregulated and also attacked by the virus. So NEMO, its regulatory element, is attacked by the virus. So the hope here is in understanding this mechanism that we can actually start to find these different therapeutic approaches. And now the next stage is to really get these different existing drugs. So these are already been through safety trials. They've been used for other conditions for years. So can we design large-scale clinical trials to test individual drugs as well as combination of drugs to try to shut down this COVID-19 mechanism? And it may well take several of these different drugs in combination to do it. You can imagine that, you know, if you're in a sailboat and you go over a reef and puncture your hull and you have 10 punctures in your hull, well, just having one cork to plug one of the holes doesn't stop the boat from sinking. You need corks for each one of those holes. So we're trying to think about combinations of drugs that would collectively shut down this mechanism. We're expanding this model, looking further downstreams, looking at other angles of this. We also have a paper under review on the coagulation disorders in COVID-19 that actually has ties into this model. And we're also working on manuscripts around the immune system deficiencies we see and how they may be connected to this model as well. Some really exciting things coming out, but now we have a strong hypothesis that seems to be being buttressed by clinical work that we can go after and start testing therapies as well as doing animal model work for more invasive experiments that we wouldn't do in humans. But we can start to do clinical trials with these already approved existing drugs. Similarly, there are drugs to address the hyaluronic acid. There's a drug that represses the expression of all the hyaluronic synthase genes so we can stop the production. And there are existing therapies that use hyaluronidase, the enzyme that degrades hyaluronic acid. That's been successfully used in therapies before. It's actually introduced those enzymes into patients. So that's another trial we would like to get underway of stop the synthesis of hyaluronic acid and externally boost the amount of enzyme present to degrade it, to try to break up that membrane that's preventing gas exchange. So this work has been published in this paper in eLife, and it's gotten, of course, a tremendous response. And Amanda referred to the article in the Medium. There's been a whole string of first articles in the scientific press, and now broader and broader in the general media audience, bringing awareness. And this is actually really helpful. This model is getting traction. We're building a lot of collaborative networks for follow-up work. We're interacting with a lot of clinical organizations for follow-up work in clinical trials. Now, in addition to this, we're also doing a lot of work with clinical data itself. We're fortunate at Oak Ridge, as part of a collaboration with the Veterans Administration, we have the electronic health records for all the Veterans Administration patients. So that's 23 million people going back 20 years. and we're actively tracking the COVID-19 patients in that population, identifying those that are either outpatients or inpatients in the hospital, and using, collectively looking at all of the clinical records, and using our explainable AI algorithms, again, just like we deal with omics data, to try to find connections within and across all of these layers of clinical information to give us clinical insight and to give us insights into the molecular mechanisms as well. Can we find associations in the clinical data that matches up with our molecular models? Really exciting work. We're starting to see some very interesting associations with our VA collaborators. We already have a paper under review, hopefully final revisions with some classic epidemiology approaches to this. And with the explainable AI and other algorithmic approaches, we're in the process of developing several other manuscripts explaining the clinical indications. We can build predictive risk models, and we can also get mechanistic understanding. We can understand the comorbidities and their relationships much better with these sort of deep dives. It also gives us an opportunity to track patients over time. So many of you have probably heard of the long hauler phenomena. Patients who presumably after they've had coronavirus and test negative for it still continue on with symptoms or even develop new symptoms. It appears there may be tens of thousands, if not hundreds of thousands of people in the United States alone that are having these long haul implications. We are working with some of these long hauler groups. It's been a really amazing experience. There have been groups, support groups that have really emerged online, often using social media to talk to each other. And they've organized in really interesting ways. And they're very interested in research, and they're highly motivated people. So there's already survey data from a couple of these different groups that's giving an insight into what are all those symptoms and in what order are they occurring. And so we're talking to these groups in collaboration. We want to use our explainable AI algorithms to really interrogate that data and to understand more of it and see if it matches our mechanisms or are there other things at play. Our hypothesis is it could be that once the bradykinin storm is out of whack, once you have a bradykinin storm, that you need to reboot, you need to reset, or it will simply continue and propagate itself. Or it could be that there is still live virus in patients. And we know for a fact that you can find virus elsewhere in the body even when they're testing negative in the nasal passages. And there was a paper just last week on finding a lot of virus in the feces of some patients who had tested negative in the nasal passages. So is there a reservoir for the virus? Is it still bouncing around the system and causing damage? And or is it simply this bradykinin storm mechanism continuing on even after the infection is cleared? All good questions to ask that we're very actively engaged in. We're also looking at environmental information. For other projects, we had to put together some massive data sets capturing environmental information. So for every square kilometer of land on the planet, we have very long, we have data sets representing lots of environmental layers. Properties of the soil, properties of the weather, cloud cover, aridity, light spectral quality, everything we can get our hands on to understand the environment. We were actually doing that originally for bioenergy, trying to understand how organisms and plants adapt to their environment. And so understanding adaptation can allow us to develop better bioenergy crops and better agricultural crops that can grow more efficiently in marginal lands and prevent us from cutting down more native ecosystems. But that work also turns out to be, of course, we as humans are also conditional on our environment, and disease states can be radically affected by the environment that the individual's living in. So we're using these same data layers we've used for bioenergy and ecosystem studies in both genetic disorders, understanding the impact of disease on genetic disorders, but now on infectious diseases as well. And it's early days. We need to get it through peer review, but we're seeing very interesting signals of environmental variables that seem to be predictive of bad outcomes in COVID-19. we've done a comparison of all these 157 million vectors on the planet, and all against all comparison. And from the result of that, we can build a network of the similarity of different points on the planet to each other and cluster that. So we have very high resolution climate type zones surrounding the globe. So for any point on the planet, we can tell you what's similar to that. Environmentally, what's similar to that region. So if we find environmental associations with bad disease outcomes, we can predict where else that might be occurring. That calculation that we did to compare all points of land on the planet was a 22 zeta up calculation. That's 10 to the 22 floating point operations. To our knowledge, that's the largest calculation done in human history. It was the second fastest. This was an exascale calculation, so we hit 2.3 exaflops of speed. That's the number of floating point operations per second. Only second in speed to a genomics calculation we'd done before, looking for interactions in genomes. That was a 2.4 exaflop calculation. So our lab and this fantastic team that I'm fortunate to lead has pulled off the two largest and the two fastest calculations done in human history for biological problems, solving big scale biological problems. So we're pumping all of this clinical and omics, as well in environmental information, as well as demographic information, socioeconomic information, housing information, policy information, hospitalization information, all into these integrated models to try to understand what's going on and generate more research and more experiments to test up these hypotheses. We're also doing a considerable amount of work, as I mentioned at the beginning, on the evolution of the virus itself. And we have another paper coming out in Molecular Biology and Evolution, in which is where we found this connection between the 3CO protease of the virus and NEMO, the regulatory factor for NF-kappa B. That's led to a number of other experiments within the DOE system. We're also working very hard on thinking about zoonosis. These sorts of viruses emerge from wildlife populations. We have seen an ever increasing number of epidemics coming out of wildlife because we're encroaching more and more on natural ecosystems. As we perturb those ecosystems, we're gonna put those systems under stress. We know in bats, when you put bats under stress, they actually shed more virus. And as you disturb their ecosystem, if you cut down the forest that they roost in, then they're going to come roost in your houses and your fruit trees and in your farms and have more and more exposure to people and have a zoonotic spillover event. So we're trying to learn about that at a global scale. We have several proposals in to do exactly that, to set up large monitoring campaigns and the sort of AI approach and global climate type approach to try to model how zoonosis occurs with the idea of being able to prevent it in the future so we don't have pandemics like the one we're currently living through. We're able to do a lot of this work because we have these fantastic compute resources at Oak Ridge, things like the Summit supercomputer and the Frontier supercomputer that's coming next. This is the largest single collection of GPUs on the planet. It also has 200,000 CPU cores, over 250 petabytes of spinning disk, and archival storage that can probably stretch into the exabytes. We also have other specialty compute platforms, big memory platforms, but this computational ecosystem that we're fortunate to be able to use allows us to dream big. When we are solving problems that I've been dreaming of for literally decades, that in the past couple of years with Summit have become feasible for the first time when we're actually doing them. Things that would have taken 340,000 years on my laptop can take 17 days on Summit, and will probably take a couple of days on Frontier, our next supercomputer. So things that were simply infeasible before, all of a sudden are reality, and allows us to ask these really sophisticated questions about biology and actually start to answer them. So big shout outs to the funding that the Oak Ridge National Lab get us from internal funding. This is called an LDRD program. It's discretionary funding that comes out of our overhead. We were able to get off the ground with both LDR and seed funding. Bruce Arno and our team was supported by the NIH. A really important point is the work we're doing builds on decades of public funding for fundamental science. Research investment in computational infrastructure like the Summit supercomputer, but decades and decades of biochemistry and molecular biology and fundamental biology. We're building on top of that work. We're interpreting our results in the context of the scientific literature and scientific data sets and databases that have come before. We're doing a lot of new algorithm development, finding new ways to analyze that data, interpret that data. But if there was no previous knowledge, this would be very hard. So we don't have to start from scratch. So, and also within our own group, the reason we've been able to pivot so quickly and hopefully start to explain what's going on in the COVID-19 pandemic is due to years of our work funded by completely other programs from the Department of Energy for bioenergy and ecosystem science, from the NIH and other agencies for biomedical science. We've been able to apply the tools we've built for other problems, refocus them on COVID-19 to pretty rapidly start to hopefully unravel the pathogenesis and the evolution and the environmental impacts, all in scientifically really rocket speed, a short amount of time. There's a ton more to do, but we think we've been able to get some interesting insights quite quickly, building on the shoulders of all the work that's come before us, as well as our own work that we put a lot of investment into it, as has funding agencies. Big shout outs to my co-authors on these papers. I lead a fantastic group of really smart students and postdoc and staff members and collaborators that have allowed us to do, I think, some really compelling work that we hope is going to make a difference. And I'm so fortunate to have these people in my team, both in my lab as well as these wonderful collaborators. Amy Justice and Joe King at the Veterans Affairs, the Veterans Affairs Healthcare System and at Yale have been just phenomenal collaborators for several years, and we've really jumped with both feet in the COVID-19, doing lots of work together. Our wonderful collaborators at the Cincinnati Children's Hospital and the University of Cincinnati, John Peschen and Bruce Arno, lead some beautiful work there and labs there. And Alan Mass, the University of Wisconsin, and the Blood Institute there has been a key collaborator in this work as well as in the coagulopathy paper that hopefully will come out in the not too distant future once we get it through the peer review system. And the list goes on. Ben Brown at the Berkeley National Lab is doing some wonderful work with us on a variety of fronts, on the explainable AI front and the iterative random forest work, as well as with other interesting environmental components that we're studying together. Really fantastic set of collaborators there. So again, in our minds, we move science forward by collaboration, both within the group as well as across groups. No one individual has what it takes to know it all and to bring all these vastly different disciplines to move the bar forward. And that's what we want to do. These are the people involved currently and historically. In my group, it's a really wonderful group of students and postdocs and staff members. Big shout outs to all the people involved in this work. And with that, I can take questions.
Okay. Wow, that was a lot of information, and thank you for giving us so much. We've had a couple of people comment that they want to possibly see your slides or watch the recording that we have again, just to make sure that they catch everything. I have a lot of questions. The first one I want to start out with is just to clarify that I understand what's going on here. You were saying that without going into too much on the specific genes, that the presence of the COVID virus causes the bradykinin to downregulate, and so that causes blood and permeability of the various blood vessels, and then also causes the hyaluronic acid to be more available, and that also can absorb a lot more water, and all of that together causes more inflammation, more fluid, that sort of thing. Am I understanding that correctly?
It's actually the overproduction of bradykinin. It's the excess of bradykinin that leads to these effects, that will lead to the permeability of the blood vessels, that will lead to the inflammatory response, that will lead to the hypotension, low blood pressure. And it's actually connections from the RAS pathway that seems to block the degradation of hyaluronic acid. And by some other mechanism, likely hyaluronic acid expression is the genes that synthesize hyaluronic acid are upregulated. We haven't, it may well be that there's a unified mechanism there that downregulating one upregulates the other. We haven't found that quite yet, but we're looking.
So have you seen in the samples that you've taken, the many thousands of samples, were both of those present in all of the samples, or did one have the bradykinin and one had the hyaluronic acid, or was it both? No, how do they interact?
It's both. We're seeing these changes concomitant. So the changes in the RAS pathway and the changes in the kallikrein pathways and the changes in the hyaluronic acid are pretty consistent across the COVID-19 patients. And the way you do statistics is you compare them as a group versus the controls as a group, and you see, do they collectively, are they collectively different? And they are, and these are pretty consistent patterns. We have other studies with other data sets we're looking at now that are comparisons between asymptomatic patients or mild illness versus severe illness, actually in similar tissues, but with single cell data. And we're looking to see, what's the transition, not just from an uninfected person, but from a mild case to a severe case. So again, lots more work to do, many more questions to answer, but we're trying to understand that at higher and higher resolution, all the way down to what's happening in individual cell types and to a degree, even in individual cells.
And you were talking about that this could help explain the diversity of symptoms from, the COVID toes and the lungs and heart issues and all of that. Am I correct in understanding that we don't have a clear understanding of why those happen in different areas necessarily? Is that correct?
Well, I mean, if there's an excess of bradykinin in those different areas, the symptoms are exactly what we would expect to see. And we know from other diseases that cause an excess of angioedema, often genetic diseases, hereditary diseases, we see these same symptoms in those same tissues. And in fact, even with ACE, we know the side effects of ACE inhibitors. We've heard a lot about ACE inhibitors. They're one of the pharmaceuticals used to regulate your blood pressure, and it does it by inhibiting the ACE gene, and that's how it lowers blood pressure. It tips that balance towards hypertension. Well, common symptoms of ACE inhibitors for people are a dry cough, which is a COVID-19 symptom. It can be a loss of smell, which is one of the classic COVID-19 symptoms. Severe ACE inhibitor sensitivity also can have similar neurological symptoms. So that mechanism where you're knocking down, which is going to lead to more bradykinin, or you're upregulating the bradykinin pathway, we know from other pharmaceutical evidence, sensitivities to drugs, as well as other conditions, map very well from what happens when there's an excess of bradykinin in different areas of the bodies, including the brain. You get the same set of symptoms that we're seeing in COVID-19 patients.
And to be clear, what are some other conditions that are not COVID related that are associated with an excess of the hyaluronic acid or the bradykinin?
The most common one for bradykinin is called hereditary angioedema. And angioedema really means the leaking of fluid out of blood vessels, edema, swelling. And so you'll find people with angioedema will have these attacks, and will have, it varies across the population because there are a range of different genetic variants that can cause it. But if you look across the population at all the different symptoms they have, many of them are very similar to COVID-19. It's a little bit different because that will be a systemic difference. That's a genetic defect, so that will affect kind of simultaneously all tissues in the body where it can be expressed. So you may well have some symptoms that are common in angioedema that you might not see in COVID-19 because we think it may be a point of infection. Where the virus is infecting, it's causing this change. So one of the symptoms of hereditary angioedema sometimes is facial swelling and lip swelling. That's a systemic effect. We do see some cases of COVID-19 that have that facial and lip swelling. It seems to be more rare. And that may be a tipping point where the disease is going systemic, or maybe they have actually infection in the lips and face. But typically, we think it's a more localized model where you're just going to get this where the virus happens to be. So you don't get the entire body profile until it moves around to different organ systems.
We've had a couple of questions about specific medications, specifically vitamin D supplements, and also people talking about taking ACE inhibitors. With the vitamin D, is there any evidence that taking that as a supplement is beneficial? I think you mentioned that there was a study that was looking at that, but is there any kind of evidence yet? And then also the ACE inhibitors, should people who are taking those be concerned?
First of all, the caveat, we're not giving medical advice here at all. This is research, and that's why we really want to do as many clinical trials as possible to prove efficacy and safety. But that said, yes, the study I mentioned from Spain showing a vitamin D analog is showing really encouraging results. There are a number of other more epidemiological studies looking at populations that typically have low vitamin D are having worse COVID outcomes. Areas of the world that have less UV light, that seems to have some effect. Those are broad inference studies. There are several clinical studies showing lower levels of vitamin D in the bloodstream with worse outcomes. And then these clinical results of these first clinical trials. There are other clinical trials spooling up. We're in contact with some of the leaders of those other trials for larger scale studies. But the first clinical studies were actually quite encouraging with vitamin D. Other drugs were Incavidin, the bradykinin inhibitor. Again, there's that brief small clinical study that's showing encouraging results, and then the dexamethasone steroid therapies that also hit our model have also been showing really encouraging results. So there's a lot of work to do. But the vitamin D seems to be showing benefits of its own accord. Now, I mean, people need to be careful. If you take horrific amounts of vitamin D, it can become toxic. The normal levels people normally take is sort of 700 to 1,000 or 2,000 units of vitamin D. People tolerate well. If you get up to 60,000 units, then you can get into sort of toxic effects. You would have to work really hard to do that. So don't work really hard to do that. I mean, take normal doses. Vitamin D is generally beneficial as a normal supplement. So there's not a reason not to take it just as a normal part of healthy living, irrespective of COVID.
Right. And I apologize. I told people at the beginning not to ask you medical questions, and then I kind of asked you a medical question. So I'm sorry about that. But we had...
did you have another question that I forgot?
I had just asked about the vitamin D and the ACE inhibitors. Yeah.
Oh, that's great. So now there have been a number of studies, and our partners have looked at this, and there have been several papers on this. There was initial concern about that. And so far, the papers that we've read don't show a major risk factor in ACE inhibitors or A or B blockers, things that block the angiotensin receptors. And to me, that makes perfect sense. If in COVID-19, the ACE gene is already being downregulated 8-fold, okay, 800%. So imagine if you have, let's say, arbitrary units, 80 units of ACE normally in your system, and COVID-19 comes along, the virus comes along and whacks it down to 10 units. Well, that's a massive change. If you throw an ACE inhibitor on top of that and it goes from 10 to 5 units, probably doesn't matter. You're so low already that the damage is done, the balance is tipped. So a little more ACE inhibitor probably won't matter. That's our running hypothesis, is the virus itself is acting so efficiently at downregulating ACE that a little more inhibition simply won't have won't have a significant effect. You're already past the tipping point. So people should actually be very careful. And there were reports in social media early on expressing a lot of speculation about ACE inhibitors and ARBs. And it's actually very dangerous to cold turkey stop those sorts of medications. So people are on those medications for a reason, to manage their blood pressure. And unless they have a physician's advice to stop taking it, they should stay on it. And we don't see any reason in our data or other papers that are coming out to go off of them, for sure.
In the research that you did, I'm curious, I think you said at the beginning that there were 17,000 samples from, I think, 1,000 different people. Where did you find those people, or where were those people found? And what can the data tell us about any kind of differences of children versus adults, or differences in gender?
So we're looking at a lot of different sources of data in this paper. Obviously clinical data, autopsy data. We spent a lot of time reading autopsy reports and clinical reports. And then these gene expression data sets. So we're fortunate, and this is what I was mentioning before, of standing on the shoulders of science that's gone before, and in public investment in science. So there's this large gene expression data set called GTEC, which was a large effort. It's a post-mortem study. So it's a whole collection of centers that collectively put tissue that people have donated. Similar to organ donation, people have agreed to have gene expression done on their different organ tissues. It's a real gift to the community and a gift to science. That's led to this collection of 17,000 RNA sequencing samples, gene expression samples. And that's a tremendous resource that now we can use new and creative algorithms to find these patterns in all these different tissues and understand how the system is normally to work. We're certainly already looking in various data sets about the effect of age, the effect of environment, the effect of gender, and the impacts of that, and how that may change regulatory patterns, may change expression patterns over time. We know that's the case. And so that's one of the other things we're starting to work on, is exactly splitting up these data sets, stratifying into different age groups, and maybe into different comorbidities, and understand those impacts as well, to become an even larger part of our model as we can project it onto the different parts of the population. And we're doing the same thing in the clinical records. So diving deep on lots of fronts.
Yeah, well, I'm glad to know that there are people making some progress on this kind of thing. So I have one or two more questions. The first question is, you were talking about how you have all of this research that you've done in the past, and you were able to pivot to work on the COVID question pretty quickly. My understanding is the paper that you published came out in, I think, July, and that seems very quick for a scientific paper that is peer-reviewed and all of that. I imagine that there's some, it was expedited because it's COVID related, but can you talk a little bit about the process of science and how the peer review works?
It's interesting in that you're right, in normal science, sometimes it can take a year between the time you submit a paper and actually it gets published. Sometimes it's 6 months. There's a big push to make that faster, but often it takes a considerable amount of time. And so there has been an emphasis on COVID-19 work to try to get it out faster. We certainly wrote this paper in April, and it took a while to get through the journal system. And you're right, getting out in July was sort of rocket speed. It felt like molasses to us, because we thought this was a really important mechanism to get out there and have community awareness of it and to start driving clinical trials. But it is a process. So you first submit to editors, and editors are the gatekeepers to journals. Editors themselves may reject the paper based on their own view, or their own view of whether it will reach their audience, whether it's appropriate for their mission in their journal, or whether they like the paper. And so there's often a process in papers where you submit it you submit it to a journal, and an editor will reject it, and you'll take that feedback and maybe refactor the paper and submit it to a different journal that may have a different angle or set of criteria. And often that happens a few times in publishing a paper. Once in a while you get it through in the first try. That's very unusual. But once it gets past the editor, then it goes out to peer review. So there will be multiple people reading this and criticizing everything that you've done. The job of good peer review is to tear the paper apart, find the holes, point out the things they think are good, but more often they'll point out the things that they want to criticize. So it's an independent view. And there too, the editor has a choice. If the reviews say this is not worth publishing, the editor can reject it then. Or you can say, okay, these are the critiques of the editor. Please address these. Either do more work or rebut what the reviewers are saying. And often there can be several rounds of that. You may rebut the editors, they may take your rebuttal and give you more feedback, and you may rebut again. It depends on the paper. So it's an iterative process between you and the editor and the reviewers, and back to the editor to you, back to the editor, back to the reviewers, until everybody's happy. And once everybody's happy, then there's a process of, you get into the technical editing on the journal side of, they do what used to be called typesetting for printing it on paper. But now they're reformatting it for publishing in typically online formats. There's still some of them produce paper, but I read the vast majority of my literature now online. I think most scientists do. And that is another set of technical editing, and you get proofs and have to review the proofs, and sometimes they make mistakes in the technical edits and you have to correct those. And so there's another set of iterations. And then usually once you pay the open access fees, then they'll actually put it online. And so that's a process we're engaged in all the time. We have lots of papers under review all the time, and always working their way through the system. And always other manuscripts. You're either doing the fundamental research or have finished that phase and are writing the manuscript and sharing it between collaborators. And that too is an iterative process. Different people may write different parts of the manuscript, and then you share it. Or these days we all work on one collective central document, whether that's a LaTeX document in an online environment or a Google Doc or something where we all can work together on a manuscript at the same time. And once everybody agrees, and you write a cover letter to the editor explaining why this is such great work and why they should really publish it and why it's a fantastic fit for their journal, that's your sort of opportunity to convince the editor and to communicate to the editor some things you may not have been able to actually emphasize in the paper, or ways to call out areas in the paper that you think are really important for them to pay attention to. It's your way to highlight what's the really cool stuff they should want to publish. And so it's a real process. And so we are getting papers through much faster now in COVID-19 for COVID-19 work than you would traditionally in the past. For a while, other work in journals slowed down. I mean, journals were affected by COVID-19 like all of us were. All of a sudden, everybody was disrupted and working from home and figuring out childcare and education for their kids, and the whole process slowed down. For non-COVID work, the journals were as disorganized as the rest of us trying to figure out life in COVID-19. And so reviews actually became slower, because not just the editorial staff, but also the reviewers themselves, who were typically academic scientists, their lives were disrupted as well. So I think things are getting back on kilter now, but we certainly saw this dip in the speed of review and other areas of science that we work in. Because, like everything else in society, COVID-19's had a big impact. It's hard to think of anybody on the planet that hasn't been impacted somehow by this pandemic.
Well, the fact that you were able to do that amount of work and get that published as quickly as you did is pretty remarkable, in my experience anyway. Very limited, but my final question is, what is next? I know you said that you have a couple papers coming out possibly later this week or next week. Is there going to be more work done on this bradykinin hypothesis and the hyaluronic acid? Are you doing that? I know there's clinical trials. What's next?
We're doing a number of follow-up studies on this and expanding the model into the downstream signaling cascades, looking further down what happens downstream from the bradykinin receptors, how that ties into more of the immune response, the coagulation disorders, the coagulopathy. That's a manuscript we already have written, and increasingly tying all those different mechanisms together. Looking at the long haulers, how does this match up to them, getting those studies moving, all the clinical trial work that needs to happen on the drugs we think we have targets for now and other drugs that will probably come out from this expanded model that we're working on. Other types of data sets to layer into this. The clinical work is ongoing to find lots of other clinical indicators and predictors and comorbidities, understanding the clinical interaction with different diseases that lead to bad outcomes, and diving back in to understand the mechanisms of those comorbidities or other lifestyle or environmental factors that may be impacting severity of disease or even transmission. Our work on the molecular evolution of the virus continues. We have a paper with a very cool analysis of how the virus is changing during the pandemic. So within species evolution, the changes that are happening during the pandemic and how that may be affecting the behavior of the virus. We think a really, really cool set of mechanisms and analysis that, again, we hope we've submitted our final revisions, but we are awaiting the reviewers and editors to see if they agree. But some really exciting work there that's pointing out, I think it'll be an interesting paper, and we think some of the existing analysis may be getting it wrong. A more comprehensive and sophisticated approach is giving us a different view of how evolution is happening that will contradict some of the analysis that's happened so far. So we're very keen to get that out because it has really interesting implications. And the evolutionary work gives us functional insights. All the mutations we're projecting into the three-dimensional protein space, and we're trying to understand the interactions between the proteins and what we can learn from that, from the natural mutations, and then designing experiments with many collaborators to follow up experimentally on that, to create crystal structures or neutron, either X-ray crystal data or neutron crystal data, or experimental assay, biochemical assay data on the mutant forms of the proteins to see if our predictions from molecular dynamics and three-dimensional modeling, to see if we can validate those experimentally. So everywhere you look, there's much more studies going on, much more work underway already, a lot of manuscripts in the pipeline, a lot already under review, and tons more work to do.
Well, I'm hopeful that we will get some answers soon that are able to be used with the public. Actually, I have one more question. The work that you're doing, how is that related to a potential vaccine, or how will that relate, just briefly?
We haven't focused in the vaccine field. I mean, there's a lot of people focused on that. That said, I mean, right now, the target for success for vaccines that would be released currently, the bar is 50% efficacy. So if you have 50% efficacy, that's great. That hopefully will slow down the spread of the virus, but that still means you're going to have a lot of sick people that we need therapies for. So, and it's going to take a while to distribute and inoculate people. And probably many of the current vaccines they're looking at will require multiple inoculations. So that's going to take time, and so we need solutions in the interim, and that's if all the vaccines work. And that's an open question. That's why they're doing all the clinical trials now, and that work is to try to prove safety and efficacy. We saw one of the vaccine trials was halted due to these side effects, and that's a big concern. We really want to make sure that safety is primary importance in the vaccine work. That Hippocrates' oath, first of all, do no harm. And do no harm, and then make sure that it's actually effective. So we're not focused on the vaccine work. We're very aware of it and tracking it. We're as interested as anybody. But we think understanding the fundamental mechanisms of this will be important. The evolutionary components of what we do will have impacts on vaccines, though, as we understand how the virus is changing and where it's changing. We hope to be able to interact with, track, and maybe even predict future mutations that may make a vaccine ineffective. And a vaccine would be a selective pressure for mutations that allow a virus to escape the vaccine. So there's, vaccines themselves will select for mutations that allow the virus not to be recognized in a vaccinated system. So that's certainly the ongoing work and the evolution of the virus that does have direct impacts on the long-term success of vaccines.
Well, fascinating stuff. I wish you the best of luck in all of your work, and I look forward to reading some more. And so I would like to say thank you to Dr. Daniel Jacobson for joining us this evening and giving us just a ton of information on this, and I look forward to finding out more. I am going to wrap up this evening by sharing my screen and just telling you a few more little things. Let's start here.
If people have questions later, I can hang out after we wrap up and answer a few more questions if people have them, or if they want to run away screaming, that's fine too.
Sure. Yeah. Why don't you stick around in the chat for a few minutes? I know that there were some more technical questions that a few people had that I wasn't able to get to. So if you want to maybe get to some of those, we can answer those. So thank you for that. I am going to tell you about what is coming up next week, next Thursday, September 24th. Excuse me, we're going to be talking about a completely different topic, understanding bird behavior, an illustrated guide to what birds do, and with Dr. Wenfei Tong, who is a biologist, and should be a fascinating talk. So birds seem to be very popular with our audience, so please join us for that talk next week. And I also want to say thank you to our 109 Patreon supporters, which is just fantastic and wonderful. And we couldn't continue to do this without you. I want to say a special thank you to one of our Patreon supporters, Carol Stewart, for being here. Hope you're still here. And thank you, all of you. These are the folks who are joining us at $10 or more per month. And we have many more at $5, and you all are wonderful people. So thank you for that. And also, finally, if you are interested in helping support Science on Tap, we are a donation-funded organization. The vast majority of our events are free and available to anyone, and we put them on YouTube afterward. If you are able to make a one-time donation, you can donate through our nonprofit partner, makeyoutthink.org/support, that website there. Or if you want to join us on Patreon, you can go to patreon.com/makeyoutthink. And thank you all for coming this evening. If you joined us on Zoom, you'll be getting a survey from us in the next day or so. We'd love to get your thoughts on this. And I am going to turn off my screen and invite Dr. Jacobson back on if he wants to do a little bit of chat. And thank you all for joining us this evening.
Okay, one question, could you talk a little bit about the XAI algorithms mentioned, what they do, how they work, and why those algorithms' explainability as opposed to the explainability of the produced results is important for your process? So one of the workhorses we use is a algorithm called iterative random forest, originally developed by Ben Brown at the Berkeley National Lab. We've done a lot of work. Some very good students in my lab, Ashley Cliff and Jonathan Romero, have done a lot of work to further develop those algorithms, add lots of new features, and to scale them up on the Summit supercomputer and other architectures, and then to invent new ways like IRF loop, which is a IRF leave one out prediction, so that we can actually take a matrix or a set of matrices and use one of those elements of the matrix as something you're trying to predict with the rest of the matrix. So you can find combinatorial predictors, relationships between objects in and between matrices. That's allowing a lot of new work. It's the foundation, not surprisingly by the name, of random forests, which have been a workhorse for machine learning for a long time. The idea of random forest is it's a forest, a collection of decision trees. And decision trees are ways of splitting up data and trying to see what portions of your matrix is predictive of a Y vector. Decision trees can be weak predictors, weak learners, and can overfit, but an interesting phenomenon when you put a lot of individual decision trees together in a random forest, they collectively become a strong learner. So it's an ensemble approach. The explainability comes in that it's not a black box. Things like deep learning are good for classification. They can recognize a pattern, but they won't tell you what the pattern is. IRF combined with random intersection trees, another algorithm, we'll find is a very good way to find not just a predictive value, a predictive model, a classification model, but to find out what are the important variables that are responsible for recognizing a pattern. And that's what I mean by explainability. And in biology, we don't just want to recognize a pattern or be able to predict a pattern. We want to know what are the important things in a data set that are responsible for that. We've done a number of studies showing that these sorts of approaches are much more accurate, much more informative than traditional data analytic approaches that have been used in the past. So these are new ways to do expression analysis and cross-omic analysis that give us an explainable model we can capture in a network, capturing those relationships between the entities of a cell, the objects of a cell. And it's that collective understanding then that we can mine for specific understanding around the whole range of topics, including COVID-19. Other questions? Let's see. Is EIA cover more? Is the ground glass phenotype reversible or irreversible? We think over time, it's actually an excellent question. It's one of the therapies that we think needs to be tried. The easier therapy is the drug that stops the production of the hyaluronic acid. But once it's there, can we get rid of it? The degradation is downregulated. There are therapies, again, developed for other diseases that have hyaluroniases, the proteins that degrade hyaluronic acid polymers. There are ways to actually administer those to patients to break down existing hyaluronic acid. So it's exactly those therapies we would like to try in COVID-19 patients in clinical trials to see, can we reverse that amount of hyaluronic acid? Is the underlying damage and perhaps possible scarring reversible? That's an open question, and that's a real concern. Is the damage to the lung tissue itself that can be done during pathogenesis, will that heal or will that scar up, and will that lead to respiratory deficits in the future? All open questions, but things we're really concerned about. Are there clinicals on the studies of bradykinin levels that support this? There's been lots of research in the kallikrein and bradykinin system in the past and in animal models that support the hypothesis under experimental conditions. In COVID-19, in patients themselves, accessing samples at the points of infection is very difficult. If we say we want to measure the bradykinin levels in your brain, can we please have a sample of your brain? That's not a very popular request. It's not an ethical request. We couldn't do it in a live COVID-19 patient. One of the other challenges is that bradykinin normally in circulation has a short half-life. It degrades very quickly in the overall body. So it is one of the studies we're planning on doing is this conversation between, is this a local model at points of infection, or at some point or in some patients, do you have a systemic effect? Does it change the bradykinin levels in your whole body? Our stronger hypothesis is it's localized, and so it's more likely going to be at points of infection. So that's harder to study in people. We're planning animal model experiments with animal models for COVID-19 that do get infected, that we can actually do those do those sorts of collections at points of infection in vivo. We're also planning to do these sorts of experiments in cell cultures where there's not an accessibility point of view. So there is follow-up work to do there for sure. But this is built on a lot of work that has been done in bradykinin and other conditions that supports it. But we certainly want to do it under infectious load in some system. We did talk a little bit about in the article, we have some hypotheses about why the virus seems to have a higher impact on men than women. Certainly mortality rates seem to be higher in men than women. One of the hypotheses of one of the genes we think is involved is on the X chromosome. Now women have 2 copies of X chromosomes and men just have 1. They have an X and a Y chromosome. And so women have 2 copies of each gene on the X chromosome. Normally, many genes are subject to X silencing. So only 1 copy of those 2 copies on the 2 X chromosomes will be expressed. The other will be silenced. And there are mechanisms increasingly understood about how that happens. One of the genes we pointed out in the paper is well known to escape X silencing. So you could well have a double dose of that gene in women, and we looked in the COVID-19 samples, and lo and behold, the women's values were close to twice those of the men. So if that's protective, that might be one explanation. We also know that there can be differences in the renin-angiotensin system between genders, and the impact of hormones on these systems are all possibilities and things we're looking at. And we think we have some interesting insights into that. It's probably a more complex story we're going to keep exploring. Very sciencey question. 6-month long hauler here. I know there's also talk about a prolonged hyperactive dysfunctional immune response in the long haulers. Can this be caused by the bradykinin storm? Recently watched you on the YouTube with Dr. Yo. I'm checking a scenario in mind. Is there evidence of excess bradykinin upregulating receptors or immune cells? So we're looking at exactly that. And we do know that excess bradykinin will go down through the arachidonic acid pathways, and that will lead to downstream signals for inflammation that will lead to changes in regulations of cytokines, and there will be feedback loops into those pathways. And so there will definitely be immune and inflammatory effects of bradykinin. And so if bradykinin continues to be overproduced in long haulers, that continued impact on inflammation in the immune system would be something that would be consistent with the model. So that is certainly one of the questions we're very interested in long haulers. We have a different manuscript we're working on, 2 of them in fact, focusing on the immune system implications of this. What could they use that would say it was partially, okay, do you have approaches to increase ACE? And the short answer to do we have an immediate solution to increasing ACE, we don't right now. I mean, the focus historically has been on inhibiting ACE rather than increasing it. It's something that is on our mind, and we don't have an immediate answer to that, but it's a logical thing. If we could figure out a trigger that would increase ACE expression with an existing drug somehow, that could be a very interesting therapy. So we're on the lookout for that. I don't have an answer for it, but it's something that we're actually thinking about and looking for. Yes, I'm ignoring the questions about the origin of the virus. There is a paper that just came out speculating about that. There's a lot of work that needs to be done to follow up on that. At this point, we don't have the we haven't had time to even look at the claims of that paper. So it's really hard to comment on that. It's way too early days to have an informed opinion on those papers or that paper. Sure. Is the coagulation of blood a concern? Yes. The coagulopathy, these very odd coagulation behaviors that we're seeing in COVID-19 patients is something with some great collaborators at the University of North Carolina, Chapel Hill, and Vanderbilt, and in Wisconsin. We've put a lot of work into working with them and their deep expertise in coagulation and fibrinolytic pathways, and how they tie through to some of the bradykinin pathways to try to understand what some of the mechanisms may be that are causing all these blood clotting issues in COVID-19 patients, which can be very severe. That can lead to strokes, that can lead to other sources of damage in the lung, that can possibly lead to myocardial infarctions in the heart. So the coagulation disorders are again very bizarre in COVID-19, and an active area of study for us and many others.
Okay, I'm gonna jump in here because our team, I think we need to finish up for the evening. I greatly appreciate your enthusiasm and all of your answers, and all of you folks who are still hanging on for more questions. Thank you for being here. If you have more questions, you can send them to me. I can forward them on to him. And yeah, let's all hope that they get some good answers for what to do with COVID. So thank you again, Dr. Jacobson, and thank you all for coming this evening.
Thank you for doing these sorts of podcasts. I think what you're doing is phenomenal. And I think science and folks in the public and taxpayers should be interacting a lot more. We taxpayers are funding science, and we as scientists need to communicate what we're doing and demonstrate that in gaining fundamental knowledge, we can actually make things better for people. So exactly what you're doing helps that mission a lot. So thank you for all the effort that I know goes in behind the scenes. It's been interesting for me to get a peek behind the curtain and see how much hard work goes into putting these on. So thanks for what you do.
Well, likewise. And yeah, we should continue talking. So I'm going to sign off. Thank you all for being here. I know it's super late for you, and yeah, we'll talk again. Thank you all for being here.